I would like to talk about some of our studies in the area of inflammation and cancer, and also introduce to you to some of the work that we started doing on what I consider now to be a major problem in the US, but it's also affecting the rest of the world, and that's the effect of the obesity epidemic on cancer incidence. And just before I get into the inflammation in cancer, just before I left, I was very happy to see that the second edition, the 2.0 version of Anahan and Weinberg reviews on cancer has officially recognized tumor-promoting inflammation as one of the hallmarks of cancer. So now it's official. Inflammation does contribute. Something that it took the cancer research community almost 150 years to recognize, since Virchow has suggested this in the mid-1800s. From work that we have done, as well as others, we know that there are at least 4 types of interactions between inflammation and cancer, and we can really divide them into 2. There are situations where chronic inflammation caused either by persistent infection or autoimmune disease or by some type of environmental or dietary exposure precedes tumor development, and by persisting for many years, inflammation can increase cancer risk, and eventually you end up with cancer. Not everybody with chronic inflammation will end up with cancer, but a substantial number of people suffering from different conditions will, and I will talk about liver cancer and inflammation through the major part of this hour. There are other situations, which I'll talk more about tomorrow, where inflammation follows tumor development. We call it tumor-elicited or tumor-associated inflammation. I'll talk about some examples for this tomorrow. And we also have to realize that cancer therapy, being usually cytocidal or resulting in quite a lot of trauma and injury, also induces an inflammatory response. And the question is whether this inflammation is counterproductive or actually facilitates the therapeutic outcome. But this is all for tomorrow. So a few years ago, when we started thinking about possible molecular links between inflammation and cancer, we have suggested, for very simple reasons, that maybe the NF-kappa B signaling pathway, by which a number of different receptors lead to activation of a protein kinase complex that we and others have named IKK for I-kappa B kinase, consisting of the alpha and beta catalytic subunit and the IKK gamma or NEMO regulatory subunit, may provide this molecular linkage because this complex is activated in response to a variety of different inflammatory stimuli, most notably TNF and IL-1, as well as byproducts of bacterial infections such as LPS, or viral infections, viral nucleic acid. And once it's activated, it leads to nuclear translocation of NF-kappa B transcription factors such as the P50 relay heterodimer, and these transcription factors activate a number of target genes, including genes that upregulate cell survival, inhibiting either apoptosis or necrosis. So inflammation increasing cell survival, and increased cell survival known to be a major property of cancer cell, was really a no-brainer to think that maybe that provides the link, but we had to test this idea, and we chose to test this in a model of colitis-associated cancer. It's a form of colorectal cancer, accounting for about 5% of all colorectal cancer cases. There's a very good mouse model for this malignancy, and by knocking out the IKK beta gene either in intestinal epithelial cells, which in this case, due to mutations in the beta-catenin pathway, will become premalignant cells, or in inflammatory cells, in this case lamina propria macrophages, we have shown that indeed NF-kappa B links inflammation in cancer. The way it works is that in the epithelial cells, activation of NF-kappa B is mostly important for upregulation of survival genes, for instance, BCL-XL, whereas in inflammatory cells, the activation of NF-kappa B is very important for the production of growth factors, and we have shown that one of these growth factors is none but the inflammatory cytokine IL-6, and in cancer cells or premalignant cells, IL-6 through the JAK2 tyrosine kinase leads to activation of the oncogenic STAT3 transcription factor, and STAT3 activation in epithelial cells again leads to further upregulation of survival gene as well as stimulating genes such as MYC and cyclin D1 that stimulate cell proliferation. And we have shown that this connection between inflammatory cells, including macrophages, dendritic cells, and T cells in some cases, which are recruited by chemokines that are produced at different stages of the tumorigenic process. Then these cells are recruited into the tumor microenvironment, or they pre-exist there, such as the macrophages, and then through production of a variety of cytokines, including TNF, IL-6, and IL-1, as shown by others, including Ron Abt in Beersheba, lead to activation of oncogenic transcription factors such as NF-kappa B and STAT3. However, inflammation is not the only thing that increases cancer risk, and one of the major factors that control tumor incidence and mortality is aging. And actually, most solid malignancies are really a disease of old age. If you look at the incidence of cancer as a function of age, we find that in young to middle-aged individuals, up to about 40, 45 years of age, tumor incidence is extremely low. Mortality due to cancer is very low as well. But then as you get older, cancer incidence and mortality due to cancer increase quite a lot. And there's been a lot of different postulates and theories trying to explain this intriguing link between aging and cancer. And at least my suggestion is that aging is also associated with increased inflammation. Many diseases of aging, such as rheumatoid arthritis, are clearly inflammatory diseases. IL-6, which I'll talk about furthermore during the talk, a very clear tumor-promoting cytokine. Its expression is increased during aging, and there's also an increased likelihood of metabolic disorders, including obesity with age. And you can see this image of the aging David being transformed due to fat accumulation and old age. So a few years ago, a very important epidemiological study was published in the New England Journal showing that in addition to cardiovascular disease and type 2 diabetes, there's a clear increase in cancer incidence amongst individuals with a high body mass index, high BMI. For all cancers overall, it's about 1.5 to 1.6-fold increase, depends whether you're looking at males or females. But then if you stratify the effect of obesity on different cancers, you find that the champion is liver cancer or hepatocellular carcinoma, whose risk is increased by about 4.5-fold in individuals with a body mass index between 35 and 40. There's also a very strong impact on pancreatic cancer, something that we have started to study as well in the lab, but I'll spend the rest of the time talking about hepatocellular carcinoma. So hepatocellular carcinoma is a rather unique cancer. It's a very slow-growing cancer. So you think by the growth rate, it's not an aggressive cancer because it takes so many years to develop. But if you look at outcome, you find that actually next to pancreatic cancer, It's the second most lethal cancer. 5-year survival is only 8%, despite advances in therapy. And that's the reason that currently hepatocellular carcinoma, even though it's the fifth most common cancer, it's actually the third leading cause of cancer death because of this high mortality. And it's been known for many years that hepatocellular carcinoma is associated with chronic liver inflammation caused either by viral infections, hepatitis B or hepatitis C, or by obesity, which can lead to non-alcoholic steatohepatitis, and some 30% of that progresses to non-alcoholic fatty liver disease. And currently, it's quite clear that the impact of obesity increasing the risk by 4.5-fold, but in the US affecting 30% of the population, is far greater than that of viral hepatitis, which in the US affects about 1.5% of the population and increasing risk by 17-fold. So I think obesity is becoming now a major factor to deal with when one thinks about different causes of cancer. So we decided to study the mechanisms associated with induction of hepatocellular carcinoma using a chemical carcinogen, diethylnitrosamine, and we asked whether chemical carcinogenesis of the liver is also associated with inflammation. And we have used the same approach that we've used to study colitis-associated cancer. We knocked out IKK beta in hepatocytes, generating what we call IKK beta delta hep mice. We thought that if NF-kappa B is important, these mice will be protected from induction of liver cancer because they will not activate NF-kappa B in hepatocytes. And much to our surprise, we found that these mice actually developed a lot more tumors than the control animals. Not only that more tumors have developed, but when we followed the survival of the knockouts, they died more rapidly and more extensively than the flock's controls. It took us a long time to understand that, and what helped the understanding was a second knockout of another inflammation-regulated protein kinase that we've been studying, P38 alpha. It's a MAP kinase. And again, you knock out P38 alpha in hepatocytes, and you increase the incidence of hepatocellular carcinoma, and you accelerate the mortality. So we looked for genes whose regulation depends on both P38 and IKK beta. We actually did not find any common gene for the two pathways, but what we found is that through different target genes, for instance, SOD2 and ferritin heavy chain in the case of NF-kappa B, and through HSP25, these two pathways kept under check the accumulation of reactive oxygen species, such that if you dismantle either the NF-kappa B pathway or the P38 pathway, you greatly increase the accumulation of reactive oxygen species in hepatocytes, especially the hepatocytes that are engaged in the metabolic activation of the carcinogen. Actually, a liver does not want to activate the carcinogen, it wants to inactivate the carcinogen, but by trying to inactivate it, you actually activate it. And some of the hepatocytes involved in the metabolism of DN accumulate reactive oxygen species, they die. Some of them die through apoptosis. You can identify them by tunnel assay, and many more die by necrosis. And you can see that the amount of apoptosis in the liver, we are looking here at zone 3, pericentral hepatocytes, is very low in the unchallenged liver. It remains low in the knockout liver, but once you give the carcinogen, there's quite a lot of cell death, and in the IKK beta or the P38 alpha, there's an increase in the amount of cell death. And in the liver, when you increase cell death, you trigger what's called compensatory proliferation. In the mature liver or even in the young liver, there's very low level of cell proliferation based on incorporation of bromodioxyuridine, but you injure the liver and hepatocytes start proliferating, and more cell death is associated with more cell proliferation. And if you superimpose the tunnel stain with the BRDU stain, you find that most of the proliferating hepatocytes are in close proximity to hepatocytes that have died from either apoptosis or necrosis. And this property of the liver explains why in one case NF-kappa B activation promotes tumor development, and in the other case NF-kappa B activation inhibits tumor development. And in the column, enterocytes are self-renewed and replaced on the average every 1 to 2 weeks. And if you injure the column or you don't injure the column, the cells get replaced anyway. So cell death does not really make a big impact on cell proliferation in the column. However, in the liver, most of the hepatocytes are differentiated, they are quiescent, they withdraw from the cell cycle, they get synchronized in G0, but then when you injure the liver, they go back into the cell cycle, and these cells can then become tumorogenic. So this led us to propose the following working hypothesis. This is a highly simplified image of the liver, composed mostly of parenchymal cells, hepatocytes. Interspersed between them are resident liver macrophages, also known as Kupffer cells. You introduce this procarcinogen, diethylnitrosamine, into the liver. It undergoes metabolic activation in zone 3 hepatocytes. In a few of them, it will react with DNA and will induce oncogenic mutations. However, these cells are quiescent, they do not proliferate, and the mutations will not be transmitted to the progeny. Many more cells are killed, and if you increase accumulation of reactive oxygen species, you kill more cells. And we postulate that the dead cells release signals that lead to the activation of Kupffer cells. The Kupffer cells produce growth factors, and we found that one of these growth factors is, again, nothing but IL-6. And then by stimulating the proliferation of residual hepatocytes, of surviving hepatocytes, including those that harbor oncogenic mutations, now IL-6 acts as a tumor promoter and starting the tumorogenic process by increasing the number of transformed cells in the liver and leading to other changes and cascades. So we found that IL-6 is very important for liver tumorogenesis by focusing on another aspect of hepatocellular carcinoma, and that's a striking gender bias in the incidence of the disease. Worldwide, it's about 3 to 5 times more common in men than in women. The same thing happens in mice. These are male mice given a certain dose of DN, and then females of the same genetic background are pretty much immune to induction of hepatocellular carcinoma. And we found that this relative resistance of the females is due to the ability of estrogen receptor alpha that is far more active in females than in males due to differential production of estrogens to inhibit, act as a transcriptional repressor of the IL-6 gene, which is, in this case, is transcribed mostly in Kupffer cells in response to the activation of NF-kappa B. NF-kappa B in the Kupffer cells is activated mainly by IL-1, which is released from dying hepatocytes, and then the IL-6 that is released by these cells act on the nearby hepatocytes through IL-6 receptors that are actually expressed on all cells, and that will stimulate cell proliferation and resistance to cell death. By inhibiting cell death, both IKK beta and p38 will inhibit tumor development, and JNK activation, which I didn't talk about, by promoting cell death, promotes tumor development in the liver. So with this, I'd like to switch gears and talk about the other major factor in hepatocellular carcinogenesis, which is obesity. So trying to understand how obesity works, we decided to model it in mice. We have done 2 simple experiments. In one case, we gave the mice the carcinogen as usual when they were 2 weeks of age. We waited a month, and then we split the carcinogen-treated mice into 2 not each mice into two, but into two groups, and one kept on low-fat diet, normal chow, and the other one we placed on high-fat diet where 60% of the calories are fat-derived. And unlike humans, or some humans, mice don't count calories, they eat by volume. So if the food is rich in fats, they just consume a lot of calories, and they eventually get obese. And we found that indeed this increases both tumor multiplicity, tumor size, tumor incidence, and just as was shown in the human studies, the effect was more pronounced in male mice than in female mice, although obesity would also increase hepatocellular carcinoma in females, but starting from a lower tumor number, despite that, the effect is not as pronounced as in males, and we still don't understand all the differences here, but it's remarkable that it's very similar to what is being seen in human populations. In the second situation, we have delayed the administration of DN until mice were 3 to 4 months of age. At this point, if you give the carcinogen to lean mice, mice kept on normal chow, it does not induce any cancer unless you combine it with a tumor promoter such as phenobarbital. But if you keep the mice for 3 months on high-fat diet and then give them the carcinogen, they will all develop hepatocellular carcinoma. So in this case, obesity acts as a very clear risk factor, as a tumor promoter, and it's absolutely required for induction of hepatocellular carcinoma. So we ask, what is obesity doing in the liver? So first of all, we focused on a group of protein kinases related to metabolism, and first and foremost, we looked at AKT. And the reason that we looked at AKT is that the epidemiologists who found this very intriguing link between obesity and cancer, what they do is they like to make a lot of correlations. And another correlation that was made is that higher levels of circulating insulin also associated with higher risk of cancer, especially hepatocellular carcinoma. And this led to the suggestion that maybe insulin, which can activate AKT, drives tumor development through activation of AKT. The problem with this hypothesis is it doesn't take into consideration the molecular epidemiology that happens in the molecular endocrinology that happens in the obese liver. The obese liver is insulin resistant, and the reason that obese people produce more insulin is because they do not respond to insulin, and you can see this also in mice. AKT activation is reduced in the obese liver, both in the non-tumor tissue and especially within the tumor tissue. And because you don't respond to insulin, your body makes a lot more insulin, but the more insulin you make, it doesn't really matter because the insulin receptor is completely desensitized in the obese liver. And one of the major causes of insulin resistance is increased activation of P70S6 kinase, and you can see that both in the non-tumor tissue and in the tumors, there's an increased phosphorylation of this kinase, which is activated by the mTOR through the mTORC1 complex, and I'll talk about this shortly. And you can see also increased phosphorylation of the S6 kinase substrate S6. So I'll talk more about the role of this pathway in a little while. We also looked at a group of kinases related to inflammation. I mentioned that Jun kinase is tumor promoting and P38 is tumor suppressing. And we find that obesity increases JNK activity both in the non-tumor tissue and in the tumor tissue. And again, this is consistent with early studies that we have done with Gökhan Hotamisligil at Harvard, showing that obesity activates JNK. There's also increased ERK activation in the obese liver, especially in the tumors. And interestingly, P38 activity is reduced in the obese liver. And then what we found was that obesity also results in increased phosphorylation of STAT3. And since we knew that STAT3 is a very important oncogenic transcription factor from our studies on colitis-associated cancer, we decided to take a closer look at the mechanism responsible for STAT3 activation in response to obesity. And we found, again consistent with other studies, that obesity increases the circulating levels of IL-6. There's also more IL-6 mRNA in the obese liver itself. And one cytokine that can stimulate IL-6 production is TNF. And again, you can see that in the obese liver, both in the non-tumor tissue and in the tumor tissue, there's a very large increase in expression of TNF. So we asked, what's IL-6 and TNF doing? I already told you that IL-6 is very important for the induction of hepatocellular carcinoma, and in the absence of IL-6, there's hardly any liver cancer at all. So one can argue that maybe it's required for basal tumorigenesis, but not so much for the effect of obesity. So we decided to take a look at the major receptor for TNF, the type 1 TNF receptor. And if you look at TNFR1 knockout mice, in this case, you don't affect the basal rate of tumorigenesis in lean mice, but in the absence of this receptor, the mice don't respond to obesity, and there's no increase in cancer risk. So, but it's important to really ask whether the mice without TNF receptor can actually become obese in response to high-fat diet. And the control experiment shows that at least until 6 months or so, they accumulate just as much weight as the control mice. But the problem with the weight accumulation is where the fat goes inside the mouse. And that actually turned out to be quite interesting, because in the absence of either IL-6 or TNF receptor, you can see that there's a major reduction in fat accumulation within the liver. So this is staining of the liver with oil red O, normal liver, very little fat. In response to a high-fat diet, the liver turns into foie gras. And that's what happens in gooses and ducks, and also in mice, and also in humans. And the liver actually turns completely white in these mice. But in the absence of IL-6 or type 1 TNF receptor, the liver is protected from hepatosteatosis, and as a result of the reduction in hepatosteatosis, the liver is also protected from steatohepatitis, which is increased accumulation of macrophages. This is staining with the macrophage marker F480, increased accumulation of neutrophils in the liver. They are both strongly inhibited in the absence of either IL-6 or type 1 TNF receptor. So we think that this reduction in fat-induced liver inflammation, steatohepatitis, is the major protective mechanism by which the absence of either IL-6 or TNF receptor protects from obesity-promoted liver carcinogenesis. What about STAT3 in the liver? We knocked out STAT3 in the liver, and the mice are very strongly protected from induction of liver cancer, either when they are lean or when they are obese. Interestingly, we found that STAT3 is activated not only in the mouse liver, but also in human hepatocellular carcinoma. And 70% of the hepatocellular carcinomas that we've analyzed, we looked at about 50 of them, contain STAT3 in the nucleus. And curiously, most of the tumors that have STAT3 in the nucleus do not have P65 or not much P65 REL-A and NF-kappa-B in the nucleus. The minority of the tumors that are STAT3 negative contain quite a lot of activated NF-kappa-B. So we found there's a dichotomy between STAT3 and NF-kappa-B activation in liver cancer, and STAT3, just like it is in colorectal cancer, is always tumor promoting or tumorigenic, and NF-kappa-B can really depend on the context. And without getting into much detail, it's all been published, we found that this dichotomy between NF-kappa B and STAT3 depends again on the ability of NF-kappa B to inhibit the accumulation of reactive oxygen species, which can lead to both junk activation and activation of STAT3. And this is mediated through inhibition of two phosphatases, SHIP1 and SHIP2, which keep the kinase that activates STAT3, JAK2, under control. Interestingly, there are quite a number of very promising JAK2 inhibitors that have been described. Some of them work very well in myelofibrosis, which is another type of malignancy associated with old age and chronic inflammation. And some of them are about to enter the clinic for phase 2 clinical trials in hepatocellular carcinoma. And I have quite a lot of hope for these inhibitors. So basically, what we found in mice is that many of the liver cancer risk factor, hepatitis virus infections, ethanol consumption, hepatosteatosis, and so on, they all have worked through a common mechanism, induction of chronic liver injury, oxidative stress, and this leads to increased production of inflammatory cytokines, including IL-6, which now has been shown to also be a marker for rapid progression from viral hepatitis to hepatocellular carcinoma in humans. So now what about tumor suppressors in the liver? So one of the major tumor suppressors is p53. It's quite often lost in hepatocellular carcinoma. And we became intrigued with one function of p53, and I think the battery ran out, and that's it can inhibit the activation of TORC1. And the reason that we became in this link is TORC1, whose catalytic subunit is the mTOR kinase, the mammalian target of rapamycin, is the major nutritional sensor in the liver, and nutrients can have effects on the exchange on the small GTP binding protein Rab, which is a direct activator of mTOR, and they can also lead to inhibition of an upstream kinase called AMPK. And AMPK is inhibited when energy state in the level is high, and when energy production in the cell is low, ATP levels are low, AMP levels are high, AMPK is activated. So AMPK is activated during caloric restriction, exercise, dieting, and so on. And once it's activated, it inhibits TORC1. On the other hand, hypernutrition leads to increased TORC1 activity. And somehow, p53 was shown to be connected to all of this. And again, being a tumor suppressor, we were intrigued, how can p53 regulate the activity of TORC1? And at that time, I was joined by a new postdoctoral fellow, Andrei Budanov, who, for his PhD thesis with Peter Chumakov, has characterized two new p53 target genes called cestrin-1 and cestrin-2. He came to the lab trying to understand what they do, and after several years, finding that they do not affect any of the signaling pathways that we studied, he found that eventually what they do is they are very strong activators of AMPK in response to transcriptional induction by p53. The cestrins accumulate, they associate with AMPK, activate AMPK, and they allow AMPK now to inhibit the activation of TORC1. And you can see in this experiment, we induce genotoxic stress. We activate p53 by treatment of cells with camptothecin, and this results in very strong inhibition of P70 S6 kinase, S6 phosphorylation. This is all due to activation to inhibition of TORC1 activity and activation of AMPK. And in cells that lack the cestrin-1 protein, you do not see these effects. We wanted to see whether this happens in the liver. We have generated cestrin-2 knockout mice, treated them with DN, which induces genotoxic stress. It's an alkylating agent. It activates P53. And by staining the liver for S6 phosphorylation, you can see that in the pericentral hepatocytes that metabolize DN and activate reactive oxygen species, there's very strong inhibition of S6 phosphorylation. And in the same group of cells, in the cestrin-2 knockout mice, you do not see as pronounced effect on S6 phosphorylation. So we started looking at effects of cestrin on metabolism and liver tumorigenesis. And when we put the mice on high-fat diet or cross them with OB mutant mice, which are leptin deficient, we find that in the absence of cestrin, mice become more glucose intolerant, and they also become insulin resistant. And when we look at their hepatocytes, they accumulate very large lipid droplets, much more than the control mice who are placed on high-fat diet. So clearly, cestrin has a pronounced effect on metabolism in the liver. There's also increased production of liver triglycerides and so on. And we are still waiting for the results for the tumor formation experiments. But the problem with the cestrin is that it's a member of a family of proteins whose biochemical function is highly conserved all the way from C. elegans to man. And cestrin-1 and cestrin-2 and cestrin-3 have exactly the same biochemical function. So we were worried that maybe the cestrin-2 knockout mice do not really give us a very accurate picture due to redundancy with the other two cestrins. And to fully appreciate what these proteins are doing, we switched to look at an organism that Ami did not mention in his introduction, and that's Drosophila. So Drosophila has a single cestrin. It's highly conserved structurally and biochemically. And a postdoctoral in the laboratory, Junhee Lee, who's an outstanding Drosophila geneticist, started looking at the regulation and function of the Drosophila cestrin. And what he found was that even though the Drosophila cestrin does contain P53 binding site, the gene contains P53 binding site, it is not regulated by P53, but it's regulated by another transcription factor, FOXO, which is also involved in metabolic regulation and oxidative stress in mammals. And what Junhee found is that there is a signaling cascade, again, going from TORC1, whose activity is actually inhibited by the cestrin, through activation of reactive oxygen species, leading to activation of Jun kinase, activation of the FOXO transcription factor, induction of cestrin transcription, and then you establish a negative feedback loop that will inhibit TORC activity. So we carried out overexpression experiments expressing Drosophila cestrins in mammalian cells and mammalian cells in the Drosophila wing and the Drosophila eye. And since the time is running short, and I just summarize these experiments, and basically there's no biochemical difference between the Drosophila and the mammalian versions of the cestrins. And then we looked at the Drosophila liver. Well, Drosophila doesn't have a liver, but the equivalent of the liver is called the fat body. And even though it's called the fat body, it does not contain a whole lot of fat. But if you knock out the cestrin in the fly, you can see again with oil red O staining, there's a very large increase in accumulation of fat in the fat body. And when we look at the overall content of triglycerides in the fly, we find that these flies have 35% more triglycerides than the control flies. When you look at AMPK and S6 kinase activity, you find about 40% decrease in AMPK, 25% increase in S6 kinase. Does not seem like a whole lot, but this is sufficient to cause all these metabolic alterations, and we can restore the activity of the kinases with a variety of drugs, most notably metformin, which is also used for the treatment of diabetes in humans. What metformin does, it somehow decreases ATP production by liver mitochondria and leads to the activation of AMPK, and it does the same thing in the fly. It activates AMPK, and it replaces cestrin, and then it leads to decreased production of triglycerides in the fly, and they become lean all over again. The other thing that happens in the absence of cestrins is mice start developing cardiac arrhythmia. And I was very surprised until we've done this experiment in collaboration with Rolf Bodmer, who is actually a Drosophila cardiologist, to even know that Drosophila has a heart, and they have a very simple heart, and it beats just like the mammalian heart very regularly. So you can see very nice beating, but this is the heart of the cestrin deficient fly, and you don't have to be a cardiologist to see that every once in a while it misses a beat. So this is typical cardiac arrhythmia, and then you can put these mutant flies on metformin, you can put them on rapamycin, either activating AMPK or inhibiting mTOR, and you completely fix all the problems. The other pronounced problem in these flies has to do with another muscle, and that's the flight muscle. So the flight muscle is a typical striated muscle in the fly, and that's how it looks in the heterozygote fly. But in the cestrin deficient flies, when they're about 20 days of age, this is equivalent to about 16-year-old human. The muscle undergoes complete degeneration, and you can see the sarcomeres are completely destroyed and everything. And again, by putting the flies either on metformin or an ACAR, which is a direct activator of AMPK, you can completely prevent muscle degeneration. So we ask in detail what accounts for the degeneration of the muscle in these flies, and we noted that muscle degeneration was associated with accumulation of defective swollen mitochondria. We isolate the mitochondria, we measure cis-aconitase activity, and we found that there's increased production of reactive oxygen species, as you can tell by the reduced cis-aconitase activity. And you can also stain the tissue with dihydroetidine, and this is a very nice indicator telling you that reactive oxygen species have accumulated. And then we noted that there's also accumulation of ubiquitinated protein aggregates. We stain the muscles with anti-ubiquitin antibodies, and you can see now massive accumulation of these ubiquitinated protein aggregates associated with increased cell death in the muscle. So what can account, one process that will account for accumulation of reactive oxygen species, defective mitochondria, failed clearance of ubiquitinated protein aggregates, and increased cell death. And the one process that defects in it can account for all of these is autophagy. So autophagy is a form of cell death, but it's really not for killing cells. It's actually for protecting cells from starvation, and it allows cells to recycle their proteins and use them for generation of more energy and synthesis of new proteins. And in the absence of starvation, autophagy is an extremely important protein quality control and organelle quality control mechanism. And one of the things that it has to do, it gets rid of defective and aged mitochondria, engulfing them in autophagosomes. And by getting rid of all mitochondria, it prevents the accumulation of reactive oxygen species due to leakage of electrons from the oxidative phosphorylation chain. So if you inhibit autophagy, you accumulate a lot of old mitochondria. These old mitochondria produce reactive oxygen species They can create a lot of damage in the cell, and they can also fuse with other mitochondria and create more damage. And one way in which the TORC1 complex is linked to autophagy is through the phosphorylation of the ATG1 protein as well as ATG13. So we have created flies with decreased expression of ATG1 to mimic the effect of TORC activation. And again, we found that if we silence ATG1 in the heart, we got cardiac arrhythmia. And if we silenced it in the muscle, we got muscle degeneration, accumulation of reactive oxygen species, and so on. But in this case, we could no longer control it with metformin or rapamycin. So we are proposing that defective autophagy can, through the accumulation of reactive oxygen species, may contribute to tissue generation, aging, obesity. It's very important for clearance of lipid droplets in the liver, and can also contribute to tumor development. And current studies in the lab are actually focusing directly on the role of autophagy in tumor suppression and control of tumor development in the liver. And since it's getting a bit late, I'll skip the last part of the talk. I just want to acknowledge Shin Maeda, who started all the work. He's currently in Japan. He's actually safe. He's in Yokohama. Lu-Fen Cheng did some of the work that I described. Hide Kamada is also back in Japan, in Hiroshima, and he's the one who looked at the role of reactive oxygen species. Toshi Sakurai looked at the role of P38. Scott Nogler was responsible for describing the gender differences in IL-6 production and the role of IL-6 in the gender differences in cancer. Gubin He did all the work on STAT3. Ik-Jun Park did the work on obesity and cancer. And the Banjan Dar, which I didn't describe his work due to lack of time, is working on the role of CD44 in liver tumorigenesis. And all of the work that we've done on cestrins and Drosophila was initiated by Andrei Budanov in the lab and carried on by Ik-Jun Park. He took it to the Drosophila system. Ethan Beer, my good friend, helped us with a lot of the Drosophila genetic experiments, and another good friend, Rolf Bodmer, helped us with all the cardiology experiments in Drosophila. And thank you for your attention.